My name is Thomas Langer. I'm director of the Department for Mitochondrial Proteostasis at the Max Planck Institute for Biology of Aging. Aging affects everyone and is certainly a global challenge of our time. As a scientist, it's exciting to understand the molecular mechanism underlying the aging process in order to identify new targets to tackle age-related diseases. We are interested in mitochondria, metabolic organelles that have been linked to aging and age-related diseases from very early on. Now, the ability of mitochondria to synthesize ATP, the currency of cellular energy, declines with age, and mitochondria are the source of damaging reactive oxygen species. But their role in the aging process is much more complex, and deficiencies in mitochondria affect diverse processes from different metabolic pathways, cellular signaling pathways, inflammation, cell fate decisions, but also epigenetic regulation of the nuclear gene expression apparatus. Even more, mitochondria are not static entities, but are highly dynamic. And we are interested in the mechanisms that drive this metabolic adaptation of mitochondria and which enables them to respond and to adapt to stress conditions and different metabolic demands. And we use a combination of mouse models, CRISPR screens in cultured cells, combined with numerous proteomic changes to really understand how mitochondrial proteases shape this proteome, how they are regulated during aging, and how we can tackle them to fight diseases and age-related deterioration of mitochondria.
At the moment, I'm developing large-scale mass spectrometry-based methods to monitor systematically the mitochondrial plasticity in an aging-related context. Therein, I specifically focus on the quantification of protein complex turnover and protein processing. Moreover, I'm building software tools to analyze such high-dimensional data and also to provide the architecture to make them publicly available.
We already know that mitochondrial function declines with aging, and one such process is the biosynthesis of coenzyme Q. It is an antioxidant whose deficiency can cause several pathologies such as neurodegeneration and cardiovascular disease. I got really interested in my time in Max Planck to understand how such a hydrophobic molecule that is synthesized in the mitochondria can actually be spread in the cell in different membranes. And I think it's a very important question, because if we can understand this basic mechanism, we can protect cells such as neurons from lipid peroxidation, which is a process that contributes to several pathologies. Thomas is a really great mentor, and I have always got a lot of freedom in choosing my field of interest in doing research with him. I really love how our atmosphere is so international, and you always feel really comfortable in doing your science, and it's not only science that you enjoy but also the atmosphere.
I think the best thing about the Max Planck Institute for Biology of Aging is a great scientific infrastructure. It's also located at the campus of the University of Cologne, which enables us to scientifically communicate with a lot of researchers from different fields.
It is exciting to address these scientific questions, but it is really fun to work together in a team of young colleagues who are excited about the same questions. We are a very international group and address these questions using a broad spectrum of methods, and we have all the possibilities at our institute and an outstanding infrastructure to use all modern technologies to get answers to the questions we are most interested in.